Okay, let's get started, everyone. So my name's Hari, and I'm happy to chair the next session. So we're moving from brain-body to mechanistic tissue biology, something a little bit different. And I'm happy that my colleague Meng is going to kick us off with her talk on metabolic signals and how they regulate longevity across the scale. Meng is from Baylor, but now here at Janelia. Thank you.
Thank you, Hari. Yeah, so it's great to be in the part of the 40CP program and have this opportunity to share with you some our work. So this photo I actually took several years ago when I first visited MLFA and saw the exhibit from Nakanishi. So looking at it, I was thinking of how the life would be like if time was encased, like those old watches and stops. So it's hard to imagine, but luckily or unluckily, depending on how you look at it, time never stops. So time is an essential dimension for living organisms. So with time, beautiful and elegant structures will be built during development and to support sophisticated and complex biological functions. Then with time, those nicely structured will be change, and then the well-organized functions will decline, leading to aging and then at the end death. And this process happens to almost all organisms, but the duration of this process or varied among different species and even among different individuals. So what could be the cause for this variation? So like the question that Aristotle asked almost like more than 2,000 years ago, so why some animals being long-lived and others short-lived? So in my group, we tackle this question at the interface between metabolism and longevity. And in particular, we are interested in the signaling role of metabolite. So as we know, those are chemical products from thousands of metabolic reactions in living organisms. So they are best known for their functions as energy fields, structure building blocks, and biochemical cofactors. But now from many work in the field, we know they are also key signaling molecules. So in my group, through the years, we discovered specific metabolite that can actively mediate communications between different organelles within a cell and between different cells and tissues in organisms and between different microbes and their host sharing the same ecosystem. And then using signal-ribodices elegans, we were able to decipher those metabolite-mediated signaling mechanisms and to reveal their importance in regulating longevity. And we focus on two major sources of those signaling metabolite, the intracellular lysosomes and the environmental bacteria. And today, I would like to focus on the lysosomal side and want to share with you some published and unpublished work along this line. So in eukaryotic cells, lysosomes are active site for metabolite production. So acting downstream of endocytosis and phagocytosis, they digest all materials taken up from the environment. And same time, through interacting with autophagy, lysosomes also mediate the degradation of micromolecules and the cellular component needed for recycling. And there is a collection of hydrolases in the lysosome that catalyze those reactions and responsible for metabolite production from the lysosomes. And several years ago, we have connected one of those lysosomal hydrolases with longevity regulation in C. elegans. So we find this lipase called lipo4 is induced by several different longevity mechanisms, including reduction in insulin IGF-1 signaling, fasting, and also germline deficiency. Then my first graduate student, Andrew Follick, revealed the lysosomal localization of a lipo4 and also its lipase activities specifically at acidic pH. He also made a transgenic strains to constitutively express this lipase only in the intestine, the major vascular tissue of C. elegans, and find those animals show in the red curve live longer than the control, show the black curve. Then he further delineates the lysosome to nuclear retrograde signaling pathway mediating this longevity effect. They find when lysosomal lipolysis is induced by lipo4, a fatty acid binding protein called LBP8 is translocated from the lysosomes to the nucleus. So fatty acid binding proteins are known as lipid chaperones, so they can bind and transport different fatty acids and their derivatives to different cellular compartments. And in our case, Andy find a specific fatty acid derivative, an oleoacetanolamine, OEA, increases levels in the lipo4 transgenic strains. And then LBP8 can bind OEA and facilitate its nuclear transport. And then in the nucleus, OEA can activate the nuclear hormone receptor complex, NHR49 and NHR80, to promote longevity. Then after finding this new lysosomal signaling, we become curious, like how OEA is generated at the lysosome. So this was not known before. So we wondered whether there are specific proteins could be recruited to the lysosome in the lipo4 transgenic strains and then responsible for the induction of OEA. So to figure it out, my former postdoc, Yongyu, who now started his own lab, adapted the immunoprecipitation-based lysosome purification method in C. elegans, and then together with two current graduate students, Max Gao and Yu Chen Guan, they profiled lysosome-enriched proteome using mass spec, and then compare wild type and the long-lived lipo4 transgenic strains. And then from this analysis, they find in the lipo4 transgenic strains, there are many more proteins are recruited to the lysosome. So this is kind of unexpected to us, and then it tells us actually the lysosomal protein composition is not static. Change in lysosome metabolism can have a big impact on lysosome protein composition. And importantly, from the candidate that unique to this group, we find several enzymes that could contribute to the biosynthesis of OEA. So now we are doing more biochemical and genetic analysis to confirm their functions. But at the same time, looking at this long list, we also got other unexpected candidates. So two nuclear porin proteins, NPP6 and NPP15, are specifically enriched at the lysosomes from the lipo4 transgenic strains. And we didn't detect them at all in the wild-type lysosome proteome. So these two proteins are at the outer ring of the nuclear pore complex. So finding them make us hypothesize maybe the interactions between lysosomes and the nucleus increase in the lipo4 transgenic strains, and then this could facilitate the signaling communication between these two organelles. And to test this hypothesis, we measured the distribution of the lysosomes in intestinal cells. And indeed, we find in the lipo4 transgenic strains, more lysosomes accumulated around the nucleus. And Max also wrote a script to quantitatively and unbiasedly measure the distribution and confirm the shift towards the paranuclear region in the transgenic strains. And then whether this structural change are important for longevity. And we found when we knock down NPP6 by RNAi, it fully suppressed the lifespan extension in the lipo4 transgenic strains. So it shows that the positional change in the lysosomes does have a functional significance. So the increased proximity between lysosomes and the nuclear pore complex may facilitate the transport of a lysosome-derived metabolite into the nucleus and then help them execute their signaling effect. So this finding kind of highlight the importance of a lysosome-derived metabolite in mediating the communication between different organelles. Then how about different tissues? So a former graduate student, Mazia Savini, is interested in these questions, and she got the first hint from the transcriptal analysis of the lipo4 transgenic strains, and she found the neuron peptide signaling pathway is specifically upregulated. So as shown here in the nerve system, the neuron peptide is synthesized as a long pre-propeptide and then cleaved and amidated into their mature forms. And for all the enzymes involved in this process, their encoding genes are upregulated in the long-lived lipophil transgenic strains. And furthermore, there are over 30 neuron peptide genes are also induced. Then we screen all of them and identify one specific neuron peptide called NLP11 mediate the longevity effect. So when it's knocked down in neurons, it suppresses the lifespan extension. But if we knock it down only in the intestine, then we don't see any lifespan shortening. On the other hand, we can also increase NLP11 levels only in neurons, and it's sufficient to prolong lifespan. However, the intestine-only overexpression of NLP11 has no such effect. So together, this shows that the lysosome lipolysis in the peripheral intestine can regulate specific neuron peptide genes in the nervous system. And this endocrine regulation is crucial for longevity. Then what could be this endocrine signal here? So Maze hypothesized that free fatty acid could be the target since they are direct product from the lipolysis. And to test her hypothesis, she conducted lipodomic profiling and found in the lipophil transgenic strains, the triglyceride species are decreased in the lysosomes, but the levels of polyunsaturated fatty acid are increased. Then are those PUFAs the target she is looking for? So to test this, she knocked down FAT3, the fatty acid desaturates that required for PUFA biosynthesis, and find in these conditions, now the induction of all neuron peptide genes are completely gone, so suggesting PUFA is required. The next, she asked whether there are specific PUFAs involved in this regulation. So she has supplied all different PUFAs to the lipophil transgenic strains with a FAT3 mutation and find only DGLA, the homogamma linolenic acid, is able to rescue the induction of the neuron peptide gene expression, suggesting DGLA as a specific metabolite that mediates the communication between intestine and neurons. But we know DGLA as a lipid molecule is lipophilic, so it will be very difficult for it to migrate on its own across different tissues. So inserting factors that could help DGLA, Maze find another fatty acid binding proteins called LBP3. So here the gray bars show you the percentage of different PUFAs in the liposome from C. elegans. So you can see EPA take the largest portion. However, the DGLA take less than 3%. However, if we profile the percentage of the fatty acid that bound to the LBP3 fatty acid binding protein, you can see now DGLA take the largest portion. However, if we look at another fatty acid binding protein, LBP2, it has a very, very little binding with the DGLA. So suggesting the LBP3 proteins do have a clear preference towards DGLA as its lipid target. And furthermore, when we knock down LBP3, the rescuing effect of DGLA is completely gone. And conversely, we can just increase LBP3 levels only in your intestine is sufficient to induce the neuron peptide gene expression. However, if we switch the lipid binding domain between LBP2 and LBP3, generate chimeric proteins, and then its overexpression does not have any inducing effect. So it's really show this lipid binding protein LBP3 and the lipid metabolite DGLA coordinate together to mediate this endocrine regulation. So I don't have time to show you all the data, but we further show these LBP3 proteins can be released or secreted from the intestine and taken up by neurons. And then in the neurons, the nuclear hormone receptor NHR49 is responsible to perceive those peripheral lipid signals and regulate neuron peptide gene expression. So in summary, I think those studies really tell us that metabolites carry crucial signaling roles across the scale in organisms. So within the cells, it can mediate organelle crosstalk. And then in organisms, it can signal from one tissue to another. And we also have data show that the metabolite derived from bacteria can actually also serve as a communication cues to the host mitochondria. And importantly, at the time scale, those signaling metabolites can control how long animals live. So I really hope in the next few years through the 4DCP program, we can fully understand metabolic communication at the whole organism levels in time and space. So I already mentioned the people who have been leading those projects, but I want to thank all the current and former members in my group, and also our collaborators and 2 graduate students are going to give poster in this symposium talking about 2 different proteomics work, and I think they are really looking forward, for the feedback from the group. So please stop by and check their work. And now I'm happy to take questions.
Okay, we have time for questions. Jifu.
Fantastic talk, Wang. I have very wild and naive questions that if we just force the lysosomes to go near the nuclear pore or nuclear envelope, will that do something? Like we do just use a molecular tether or something to grab it there?
So that's a good question. We didn't try the way you mentioned, but we have been trying to manipulate a lot of factors that people already show in the cell culture system that can drive lysosome close to the nuclear. Unfortunately, I think that's why we need a 4DCP program. When we manipulate those factors in living organisms, none of them actually drive lysosome to the nucleus. So I think it really shows some of those factors that are very important in the cell culture maybe not necessarily played similar role in tissues. But we didn't try the way you mentioned. So, yeah.
Okay, Danielle?
Harry need to work on that.
Hello? Okay. So extended fasting or fasting in general has been shown to help with survival of different organisms when they're put in those conditions and longevity. Normally when that happens, the lysosome actually moves in that direction. So I'm wondering if this pathway is only for this type of fasting events or is something general that operates that way?
Yeah, so I think exactly as you mentioned, I think multiple systems show actually fasting can drive lysosomes to the paranuclear regions. I think there are also data show that actually by inhibitory mTOR, it can also drive the lysosomes close to the nucleus, but it maybe goes through similar mechanism because fasting and inhibition of TOR. And then interestingly, actually, in some of the lysosome storage disease conditions, people also see paranuclear accumulation of the lysosomes. So this seems like kind of contradictory to each other, right? So we still don't know. I think maybe it's different. Mechanism regulate the positioning under different conditions. So I think better understanding the physiological and the pathological drive for the migration in vivo, I think is very critical. Yeah.
Hi, nice talk. This is also a question about lysosome nuclear perinuclear localization. Is so interesting. I was wondering, do you have any idea what's the potential mechanism of this localization? It's like microtubule, any cytoskeleton related protein, mechanism?
Yeah, short answer, we don't know. We are doing more screens to look for all the factors that was known before to regulate the positioning of the lysosome and try to see which one plays specific role here. Yeah, it's a great question.
In the spirit of trying to understand the simple rules here behind this, I wonder what you think of if there's a connection between the fact that in your system, lipolysis, free fatty acid signaling is contributing to longevity, and the fact that lipolysis happens in mammals, typically in the fasted state, maybe in the calorically restricted state, which can also, contribute to longevity. Do you think there's a commonality there between this specific mechanism and the kinds of things that are seen in caloric restriction or intermittent fasting or whatever?
Yeah, so I do think there is a common, this is a common mechanism there. So I think that's why in the caloric restriction or even fasting condition, we see the systemic effect, right? It's a whole body benefit from it. So it means that there must be body, like organ-to-organ communication. So what could be the signal, right, to mediate? I think when we go higher organism, maybe there are more things involved, but I think the lipid itself, because it's a direct outcome, right, from fasting, I feel it's reasonable to hypothesize they will be the signaling molecule that mediate the tissue-tissue communication and to give the beneficial effect on the whole organisms. But yeah, I hope my colleague in higher organisms can check more. Yeah.
We should move on in the interest of time, but let's thank Meng
Yeah,
again.
thank you.
Next up, we have our very own Jennifer, who will be telling us about organelle dynamism at different spatiotemporal scales and why it matters.
Great. Thank you all for being here. I'm really excited to share with you some of the sort of the perspective that my lab takes in this world of 40CP. We actually focus on organelles. And if you remember last night, Ruslan's talk, he had this sort of hierarchical level of organization that I thought was very exciting and a big question mark between macromolecular complexes and cells. And basically, I'm probably not alone in that the sort of organizational connection, I think, is subcellular organization, the organization of cells into membrane-bound organelles, as well as membranous organelles. That's a key transitional organizational level in this hierarchy of levels in which material is being organized to create from molecules up to organization. Okay, well, what are these organelles? All eukaryotic cells have 9 different subcellular membrane-bound organelles, and they're listed here. There's varieties, early endosomes, late endosomes, autophagosomes, etc. But what these organelles are doing is allowing the collective activity of cells, which includes secretion carried out by the ER and Golgi, uptake and degradation of macromolecules, as we just heard from Meng, the endosomal lysosomal systems playing a key role in that. Mitochondria drive energy production. The nucleus is sequestering DNA replication and storage. Peroxisomes are involved in detoxification, and lipid droplets, which you'll hear more about later in the talk, are playing a really important role in fat storage, which ultimately can feed back to mitochondria to drive energy production. Now, my lab's been interested in trying to really look at these organelles in their spatial dynamic state inside a cell. Our thinking is that it's great to see the form of these organelles, but you also need their dynamism in order to infer function, and that really requires being able to visualize these subcellular structures. And we've been doing that using a variety of different technologies that are available here at Janelia, which include the lattice light sheet microscope, where you can very quickly, 3-dimensionally interrogate through a cell fast with roughly isotropic resolution. We've also been using grazing incidence structural illumination microscopy, which allows us to very quickly interrogate different organelles, cryostructural illumination microscopy, and focused ion beam scanning EM. Here are some of the applications, sort of in a nutshell, using these different technologies. For those of you who are interested, all the people in my lab who are using these technologies are presenting posters so that you can sort of stop by and take a look. This is, we're looking at the individual molecules as they move within different organelles, like the endoplasmic reticulum. We're looking at organelle interaction, the interactome among organelles. And we're using this really amazing technology, volumetric FibSim, to look at unusual organelles like ER exit sites that have not really been characterized in an ultrastructural 3D context in a way that provides deep insight into the way the system is forming. So what I want to do over the next few minutes is to really share with you some unexpected findings using these types of technologies to focus in on organelles and how our findings are relevant for the 40CP effort. So first I want to talk about RNA granules and their relationship to lysosomes. Basically, by peering into cells, Yacheng Lao in the lab made a very interesting observation, and that is that if she was imaging lysosomes, which you can see here are moving along microtubules, shown in green, RNA granules, shown in red, which she could induce using stress, arsenate, or heat shock, basically are hitchhiking on the surface of these lysosomes. Now, let me just say something about these RNA granules. These are not membrane-bound structures. These are simply RNA molecules that are being brought together through their interactions with proteins that have low complexity domains. Those proteins form phase condensates, and that's what those green structures are. They're basically phase condensates with carrying a lot of mRNA. And that's really important for cells under stress because it protects the mRNA that's sitting in that cytoplasm under stress conditions. Well, when Yacheng sort of dove into trying to understand the mechanism for this association between these two different organelles, she discovered this protein, annexin A11, which has a very interesting feature in that, on the one hand, it can phase partition into an RNA granule because it has a low complexity domain at one surface of the molecule. On the other surface of the molecule, it has annexin repeat domains that are calcium sensitive and draw it onto the surface of the lysosome, especially in response to calcium. Now, the lysosome, we know, is the fastest moving organelle within the cell. Molecular motors can zip it around the cell, tracking on microtubules. So this is really a wonderful way not only for the RNA granule to sort of position itself within the cell, but also to potentially move where the lysosomes move. And this turns out to be extremely important in neurons because RNA granules are the mode by which mRNA molecules move down axons to deliver mRNA to the axonal growth cone for translation, for local translation in that region. And what you can see is that if we do double labeling of cells with LAMP1 is in red, the mRNA is in green, you can see that these guys are co-associating with themselves as they track down this axon. So this is playing a very important role in how RNA granules and mRNA gets distributed within the cell. Now, it turns out that there are mutations in annexin A11 that give rise to neurodegenerative disease. And so Yacheng was interested in knowing what happens to these RNA granules in these cells. And sure enough, when you disrupt this interaction between the lysosome and the RNA through these mutations in annexin A11, you no longer see the co-trafficking of the RNA granule, labeled here with G3BB1 in green, and the lysosome shown here in red. They are separate. They go their separate ways. And we think that this could play a very important role in the underlying some of the phenotypes seen in ALS, where you have basically you're not going to have appropriate delivery of RNA to the growth cone to allow for local translation. This, again, was really only possible through really interrogating these organelles through live cell imaging technologies. And I want to move to another situation that I think is that's relevant for 40CP, and that is organelle redistributions under cell starvation. So I just want to give you a sense of the way cells are surviving. This is a well-fed state where basically you have two pathways by which the cell can get energy in the form of ATP. Glycolysis through input of glucose, but also the cell is continuously making lipid droplets for any kind of excess glucose coming into this system can feed off to basically the cell can store excess metabolites in the form of fatty acids and triglycerides in these lipid droplets that are basically storage depots for use under conditions when glycolysis is blocked, when you don't have glucose. If the cell is stressed in some way, glucose transporters are downregulated, or some other type of situation where there's no glucose available for the cell. Under those conditions, the cell only survives because these lipid droplets can now become the source of fatty acids that can feed into the TCA cycle to drive oxidative phosphorylation. And it's amazing that actually, as long as you have these lipid droplets, you can really drive some sort of kick-ass activity in that mitochondria, because the mitochondria is very efficient at making ATP from these fatty acids. So how does this, given these different pathways, our question was how the organelles that are involved in this rearrangement, this organization, how do they change under starvation conditions? In order to do that, we made use of a variety of different imaging technologies. One of them is shown here, the lattice light sheet multispectral imaging approach that we developed in collaboration with Eric Betzik's lab. Basically, we can introduce 6 different fluorescent proteins tagged at 6 different organelles, and then through multispectral unmixing, we can identify all of those organelles and watch how they distribute over time. We can also look at the association of these different organelles because we can segment out them. I mentioned that lattice light sheet has roughly isotropic resolution, so we can basically segment out these organelles and look at their interactions with each other, as shown in this interorganellar interaction map. Now, one of the things that you can see is that mitochondria are making very significant contacts, not only with the ER, but also you can see interactions with ER lipid droplets themselves. Now, here's a movie where we just segmented out individual mitochondria, and you can see color-coded up here all of the different organelles that are actually making contact with that mitochondria at any particular time over this time period of imaging. And in this case, this is a well-fed cell. Well, what happens in a cell that's starved, in particular glucose starved? And with work that I don't have time to really go through with you step by step, what we found is that lipid droplets become very closely associated with mitochondria under these conditions. More significantly, lipid droplets proliferate under starvation, glucose starvation conditions. They proliferate because autophagosomes are actually engaging in autophagic degradation of membranes, endoplasmic reticulum, whatever membranes they can get their hands on. These autophagosomes deliver these membranes to lysosomes that then release the fatty acids held in those membranes. Those fatty acids then get packaged into lipid droplets, and they are then delivered to that mitochondria through a repositioning of these lipid droplets in close proximity to that mitochondria. And that then drives this beta oxidation pathway whereby fatty acids are broken down into a form that can be fed into the TCA cycle for ATP production. Now, coupled with this mechanism, we see that mitochondria are proliferating under these conditions. We see mitochondria undergo significant fusion. They become highly tubulated. We also see them increase their surface area. If we interfere with the ability of these mitochondria to proliferate, and we can do that by knocking down the machinery that allows fusion of these mitochondria, what we now see is a massive buildup of these lipid droplets and ultimately release of fatty acids from these cells. And in fact, if we co-culture a cell that has a knockout of this mitofusin, where we see these large lipid, we see these accumulation of lipid droplets, and we can actually label these lipid droplets. We can then look for the extent to which we get fatty acid transfer to an acceptor cell. And you can see that it goes up dramatically when we interfere with mitochondrial fusion activity. Now, this turns out to be extremely relevant for neuronal cell biology because neurons have streamlined metabolic systems to optimize themselves as a circuit relay system. They don't do normal, they don't have really a good response to starvation. Instead, they're cooperating with surrounding astrocytes to deal with energy demands and metabolic needs. So, for instance, an astrocyte, which is is basically a major part of your brain, has the ability to form lipid droplets. Basically, excess glucose coming in, you will drive the formation of lipid droplets. We don't see that in neurons. Neurons are not forming lipid droplets very efficiently. We can see that the neurons can actually, what do the neurons do if they don't form lipid droplets, but they're accumulating fatty acids in response to excess glycolysis? What they do is they hand off those fatty acids to other cells. And this is a co-culture system between neurons and astrocytes where you can see that the lipid-loaded neuron will dump its fatty acids into the astrocyte, and this is the accumulation of that lipid marker in this co-culture system. This turns out once the fatty acids are taken up by the neighboring astrocyte, you can see that it is the fatty acids delivered ultimately into the mitochondria. So here is in green is mitochondrial marker. The red is the C12 lipid droplet marker. So we got lipid droplets that are docked next to the mitochondria, and you can see that that fatty acid, which is the red C12, is distributing itself throughout the mitochondria. So the mitochondria is basically using this as fuel. So this is just a summary of the results that I just described, and as well as other data that I didn't have time to tell you. But basically what we're thinking is that when neurons are stressed or starved, if you will, of glucose, which we think can happen under hyperactivity because basically the system gets overcharged with fatty acids under these conditions. Because these fatty acids can't effectively go into lipid droplets, they're used instead, they're shoved instead into lipoprotein-like particles that the neuron releases and ultimately can be taken up by neighboring astrocytes. Those astrocytes then deliver the fatty acid that is carried in these lipoprotein particles to lipid droplets that ultimately drive energy production in that mitochondria. And we think that in the case of the astrocyte, this could actually feed back onto the neuron. ATP released might act as an inhibitory trigger to quiet down these neurons that are undergoing this hyperactive response. And whenever I see Misha's movie of overactive neurons, then suddenly the astrocytes going wild, this is totally what I think of. So with that, I just want to end by saying we're hoping that we can, as part of the 40CP cell physiology, really think hard about organelles and the way that they are coordinating different larger scale cell responses to allow for this relationship between organelles, cells, and tissues and physiology. And with that, I want to thank my lab. Please go see their posters and the people responsible for the work that I just mentioned. Thank you.
We probably have time for like one question, please. Can somebody pass her the orange?
That was awesome. I have a lot of questions, but I'll just stick to one. So I want to understand the last part of the right here. Yeah. So basically, now the astrocytes are going to basically get energy from their mitochondria, but then how do I see your point that they can then be releasing signals back to the neurons, including glutamate, but like their mitochondria still is in trouble, right? Like how? So I just want to understand.
So their mitochondria is not in trouble because their mitochondria knows how to deal.
I see.
They are not their metabolic system is not streamlined in any way.
Okay.
So their mitochondria tubulates. We see it
okay.
expand and it tubulates. We see the lipid droplets accumulate. In fact, I think I have a slide here. Well, forget it. There's not enough time. But basically, we can see accumulation of lipid droplets in these astrocytes when we stress.
Right.
For instance, we do a peel strip of the brain. You see a huge accumulation of lipid droplets. They're in astrocytes and microglia.
Okay. The last thing to say is microglia is probably a major player here, and we can talk later about that.
Exactly.
Yeah, thanks.
Okay, let's thank Jennifer again. Let's move on. Next up, we have Allyson Sgro, a new group leader here in 4DCP and computation and theory. Is that? Yeah.
That's correct. Great. Thank you so much for the introduction. All right, so I'm going to change gears a little bit. Part of what I find exciting about conferences is talking about data you can't see anywhere else that's not published. So I want to talk about some work that's brand new in the lab. So there's no answers here. I'd also like to highlight up a little bit of technique development. I think, I often like to say science is the art of the possible. Sometimes our job is to make the impossible possible, and sometimes our job is to go ahead and do the experiments we can do. And so I think that this system that I'm going to talk about today here in microbes is one that really highlights the power of things that you can do outside of the body, looking at other cells in the wild. So I'd also like to give you an introduction to how I think about the world and cells. I think about cells as computers. I'm going to show you this video twice. So the first time I want to give you sort of my computational perspective. We have here is a bunch of cells. All right, they have started to starve. And what you're seeing inside is their thoughts, or what you will as soon as PowerPoint decides it's going to play this video. You can do this anytime. There we go. All right, what you're seeing inside is their cellular thoughts about their interactions with one another. Right? What they're doing here is making a life or death decision. If they don't band together, they're going to starve to death and die. And so they need to communicate to find out where other cells are and synchronize their gene expression to begin a developmental program. All right, this is starting a process that's going to allow them to make these fruiting bodies. All right, that allow them to subsist for up to 30 days in the wild, outside in the soil. This gene expression program you're watching them make decisions about is going to help them turn into two different cell types. About 20% of them will become stock cells that actually die to carry the other 80% of cells to somewhere with more food. Right, that's going to allow them to propagate. Right, this is an incredibly important computational program. And something I find magical about these kinds of cell computations is that it's not sufficient to identify just molecular players or look at a genome sequence. We really need to appreciate the dynamics of what's going on inside a cell to understand these processes. And I think this kind of question is really at the heart of 4D cellular physiology. Right, so if you're a cell, right, you sense inputs. Right, inputs are things in the environment. This can be things like mechanics, biochemical signals from other cells. All right, and you take those inputs and you encode them in yourself in some sort of computation to make a decision about what to do next. All right, and those decisions become the outputs you make. Right, these can be things like I secrete something to another cell and send it a signal. I decide to move. Maybe I turn on some gene expression to become a different kind of cell. All right, and it's these cellular level decisions that are part of the multicellular behaviors we care about. These are things like signal relays, right, that can propagate and synchronize behaviors across a population, or cause whole groups of cells to move. All Right, and it's these multicellular behaviors that build up the emergent phenomena that we care about, I think, in 4D cellular physiology, like development or tissue repair. All right, so I think this is a really central, critical question that we really need to think about as we approach this new research program. And what I'd like to talk about today is one of the technique problems I think is really important to think about. This has come up. How do we think about cells in situ? And how we do this in a way that we can bridge spatial and temporal scales and connect signaling dynamics all the way to emergent behaviors? All right, part of the way we tend to do that is by looking at laboratory cells that are very easy to manipulate in laboratory environments in a dish. And what I'd like to talk to you here today is about how are ways that we can develop new, native, interesting environments to understand how cells sense different kinds of inputs in to better understand what native outputs look like. My lab also is really interested in how native cells work and is working towards putting those cells in native environments to understand those kinds of computations. We take a joint theory experiment approach, but today I'm going to focus on that technique development. So I want to show you that video I showed you again at the beginning, but I now want to sort of walk you through exactly what's going on here biologically. All right, this is Dictyostelium, and Dictyostelium encode their starvation state to coordinate motility and synchronize gene expression. So what you're seeing here is a system where they're encoding information internally in cyclic AMP, and as they starve, they release cyclic AMP to other cells. All right, and what you're seeing are those little, in red are those little flashes of cyclic AMP. Those are internal spikes in cyclic AMP level that they then send to the next cell. Are they elongating chemotax when that happens? And actually, that also turns on gene expression programs that they synchronize. All right, so when you're seeing red here, you're seeing high cyclic AMP. Yellow is low cyclic AMP. All right, and this is the very beginning of the starvation process. So this process usually starts in laboratory cells that we can genetically manipulate around about 4 hours. And it can take between 8 to 16 hours, depending on cell density. All right, and then afterward, they're going to go on and make those fruiting body structures. So this is a process that's been pretty well characterized. People have looked at Dicty in the lab for over 100 years now. And you don't even need fancy genetic tools to look at this. All right, people knew that this process happened from looking at basically dark field microscopy. If you put cells on a plate, you'll see these beautiful waves emerge. All right, and that those waves, at the center of those waves, is where those fruiting body structures will emerge in the lab. All right, but I told you this is a wild process, right? This happens out in the dirt. If you're a big hiker, you might even see these on little pieces of mulch. All right, how does this process happen when you need to coordinate a complex environment and not on some nice, flat 2D surface? Right, that's something we'd really like to understand. So how do we get from this synthetic agar to understanding better how cells interact with and are affected by their environment? So key features we need to have both to work in the lab, but also to recapitulate natural environments. So synthetic soil in the lab hopefully is transparent, all right? We are visual creatures, and many of our tools are visual-based tools or use light, and so we want to be able to see through optically. We'd like a way to visualize particles, right? It's important to know if cells are not going somewhere because there's a particle there or just because it happens to be empty space that they're avoiding for another reason. We'd also like a way to visualize cells. I really think about computational behaviors, so I'm excited to visualize computational behaviors and see changes. But one of the challenges here is that in a lot of transparent systems, we have a lot of different boundaries, and so we need some sort of way to get around how do I deal with a refractive index change, right? If I'm shining light through and it hits an interface, it might bend, and so if I have too many interfaces, that's going to be a problem. So it's a problem I need to solve. I like a system that's survivable, right? We can make a lot of different substrates in the lab, but I need somewhere that cells can survive. So this is somewhere that's aqueous, it's got an appropriate pH, or I can control that pH. I can make sure it's oxygenated, keep it humid. And I also want somewhere where cells basically behave naturally, that they can grow, they can move, they can divide, distribute themselves the way they might in the wild, so I can better understand how they be coordinating and making decisions in the wild. I also think there's one more key property that's really important about soil that I don't want to forget, and I think it's in synthetic soil should have a tunable particle size. So if you look at soil, it's often made up of particles of different sizes. And from the point of view of a cell, this is actually be incredibly important to dictate your behaviors and how you're able to synchronize as a group. So just to sort of call this out a little bit, I think there's sort of 3 different regimes from a cellular point of view. Right? If I have particles that are similar to my own cell size, soil basically acts as a defect in my ability as a field to coordinate and propagate waves. So this is pretty, this would be pretty small soil. We're talking soil grains that would be, say, 10 to 20 microns. You can get soil and sand that small. A lot of our soil is actually a little bit bigger, has intermediate-sized particles. Right? If you can think about it, if you're a little bit bigger than cells, soil is creating channels that cells can move and signal through, and so it's actually creating quite an interesting boundary condition for cells to behave in and coordinate through. a lot of time, actually, soil can have particles that are much bigger than a cell size, hundreds of microns. And so in this case, soil is creating large irregular regions that cells can move on and also sometimes potentially move between, depending on what other structures are in there. But in this case, you're basically getting large irregular regions that cells can now coordinate in and crawl around in 3D. And so that's also going to be a very interesting question to consider. So we were looking for a solution to this problem. We found it in this mineral cryolite. This is something called ice rock. If you'd like to buy this, it's available on Etsy. It's supposed to give you good vibes. I think it gives great vibes. Since we started having it in the lab, I got this job, so things are working great. But what's really cool about it is that it has a refractive index that's very, very, very close to water. All right, so that interface problem I told you about, mostly solved, and we can look through it. And so that we're able to actually see and use a lot of the tools that we've already developed, right, to use on 2D substrates in this 3D system. So very exciting. So what I want to do for the rest of the talk is show you examples of this working and all the different things we're able to see. In these videos, I want to sort of give you a code of sort of optically what you're seeing here. Dark space is cryolite, right? This is a mineral. We don't dye it. All right, so anything that's dark, that's a rock. Blue is going to be the media. All right, dictyostelium live in the woods, so we use a very minimal buffer, sort of like rainwater-esque, and we dye this with a little bit of Alexa fluor so we can visualize it in this system, so it'll be blue. And then we're using the same cells that I showed you before, right, where we have an intracellular cyclic AMP reporter, because in long term we're particularly interested in cellular computations. So you're going to see that yellow low, red high code to sort of code where cells are. But if we're looking at cells in development, they're going to appear just yellow. So early on, I wanted to see if this was working. So what you're going to see here, this is a XY slice, and then you can see the sort of ZX and ZY on each side. So you can watch cells crawl. They're able to move and both around, up and down and around these substrates. This is only 15 minutes. Dicty are pretty active. That's why you're seeing a lot of movement. So let's go ahead and zoom out. How long can we look at these cells for in these microcosms? So this is a photo of the microcosm in bright field, just to give you a sense. Scale, these things are millimeters in size. This particular one is using rather, not gigantic particles, but basically a few hundred microns in size. All right, we can tune particle size by basically filtering and sieving out the way you would if you were a miner. We can seed cells in these microcosms. So initially, you're seeing very few cells. But if we also put bacteria in there, we can grow cells in situ. All right, so they can actually naturally feed and grow up over the course of a day. So now all those yellow particles are the cells ready to starve. They've run out of food, so they're going to initiate signaling. All right, so you see those high cyclic AMP waves propagating throughout the whole population. All right, and those waves are inducing aggregation along where you saw those centers begin to form. All right, we can induce this in multiple particle sizes, so I'll show you data with both very small and very large particles today. With some larger ones, we'll see a very similar process where you have very few cells, grow them up over the course of a little more than a day. They'll begin to starve. Again, you'll see these huge signaling domains that span the whole microcosm, and these will also drive aggregation. Something we find really interesting in these very large particle situations is that the cells do really like to appear as aggregate on top of these particles, or sort of on the sort of corners where these particles seem to be contacting the glass. So it's something we find very interesting. We can, if we have a really dense area, particularly if we have smaller particles, reproduce those population-weight spirals as well. So I've been showing you snapshots to give you a sense of size of how big these things are, but you can, depending on where they start, actually get whole microcosm-wide spirals. So those behaviors that we saw across populations previously, even in 2D, are still preserved in 3D. We also have the ability, because we have the ability to tune how much food we put into a microcosm, we can tune cell densities and mimic different kinds of environmental conditions that we might see in the wild. So these are two different microcosm experiments that were seeded with different amounts of bacteria. So they've just started to starve, and you can see those waves start. If you have many, many more cells, many more of the cells will be able to aggregate in the first round of aggregation. In these other systems, we have basically many cells are left behind. And so it mimics natural systems where you end up having multiple second and third rounds of aggregation because cells are not able to synchronize when you have not just defects, but too much empty space. This is truly a signal relay system, right? It is a diffusible small molecule, but if you're not amplifying the signal at each step, it often drops off quickly. Just to show you a few more cool behaviors that I think are really exciting. Again, this is a natural system. Not everything is perfectly synchronized the way it is if you starve something synthetically in the lab. So here in this video, now we're stepping up through the microcosm, so you can see different layers of depth. Thank you to my students for helping animate this. As you're getting darker, we're going farther up into the microcosm. But what you can see here on the very top is that you can have a starving stream of cells that's signaling and coordinating. It's drawing in cells from the local area. Yet down here on the bottom, we still have cells that are feeding and appear to be dynamically crawling around. So we see a real diversity of natural behaviors and how things synchronize. And I just want to sort of highlight some cool details of that 3D branch structure. Right, so this is in fact has thickness. Right, we can see this stream over about 20 microns of depth. And you can also see that it will split up and come back together over small particles. All right, so they don't, the defects that you have in the environment don't actually split things up and cause new centers to form. All right, but there's something very attractive. Once you've decided to go in a particular direction, you're going to keep going over there. Sometimes these aggregation centers can be incredibly locally attractive and not interact. So a good example here, right, this is only 100 microns. These are quite small aggregation centers. And even though they're a few hundred microns apart, and if we were in a dish, we would see these two interact and possibly attract one another. Here, they're each drawing from very local areas and have no crosstalk. And finally, I want to show you my favorite behavior. This is particularly exciting. So in this microcosm, you can see a lot of the cells are sort of weaving up and down over different particles. And they're sort of pushing some of them out of the way a little bit, right? That stream makes some movement. But if we want to zoom in, and I'm going to go ahead and do this in some details over here, some of these cells actually grab onto particles and remodel their environment. So I find this particularly cool. We're not sure if this is just native cell stickiness or an active behavior where there is some sort of active remodeling going on. Dictya are pretty amazing. They farm their own bacteria, so it could be reasonable that they have mechanisms to do some sort of environmental reshaping, maybe creating areas for them to aggregate on. We're not quite sure, but something we're really excited to look into a little bit more. All right, so just to summarize for today, what I've shown you is that we really understand this process that happens very well in 2 dimensions. And what we developed today is a technique to look at how it happens in realistic 3-dimensional systems. Part of what I love about Dictya is that we can ask and answer questions that aren't possible anywhere else yet. All right, so we have a really high level of control experimentally in this system. All right, I've shown you that we can see signaling. We have the ability to do this kind of visualization using natural cells from the wild. There are hundreds of natural isolates available. We have many mutants available to do this kind of study and to understand how that's key for them to interact with their environment. And we have optogenetic control over this process. We can actually induce aggregation. And so that's really a powerful tool for us to see how cellular computation and the environment have interplay with one another. My lab also does a lot of modeling, and so we're really interested in how do these kinds of biochemically coupled systems coordinate in complex environments. Our existing models do work to sort of recapitulate this, and so we're very excited to see what are predictions that we can make, then test in this system. So with that, I'd like to wrap up and thank my team. I particularly want to highlight that this was all the work of Dr. Emily Hager, a postdoc in my lab, a real star. I'd also like to call out that 2 of my students have posters here. We don't just work on microbes. We're very interested in organoids. My postdoc, Brianna O'Reilly, was showing her poster on that work. We also do theoretical modeling and have some work in bacteria that Mark will be showing off. Like everyone else, I'm hiring. So if you're interested in answering these kinds of questions in both in vivo and in situ in microbes, please come talk. And with that, I'll take questions.
So Allyson, presumably the cells that become stalk versus cells that become spores, there is a competition between them, right? And the low fitness cells presumably will become stalks. I don't know if that has been directly tested, but it's presumably
people have absolutely looked at the idea of fitness and who they're competing for stalk and spore.
So given that, and that you can mark the cells based on fitness in some way, like you mark them and then you change the fitness by inducing some stress, mutations, and so on, then you can ask what controls the what reports on fitness, which fitness is important. So in mammalian cells, for example, mitochondrial fitness and mitochondrial quality and genome quality seem to be critical. But in free living cells, I'm not sure it's known, but that would be an interesting screen to do to identify what that is and how they communicate to each other, their fitness level.
I absolutely agree with you. Something I'd actually like to throw out about fitness is that people in Slimal thought they had fitness solved in a great system for all these questions about cheaters and competition. And so they actually have cheater mutants that are better than others at getting into the spore. Huge part of the field. I have a colleague who is also very interested, and she's actually putting out some beautiful work this year that shows that actually everything we thought we knew about cheaters being genetically encoded is actually totally bogus. That if you change their pre-feeding conditions, things that were more successful previously and had better fitness behave totally differently. So the fitness question is now, I think, reopened in the sense of what even controls that. If we get better traction on, again, how we could control that and what leads to that, I'm with you. It would be really awesome to look at. But for those of you who are thinking that fitness was solved, please check out that work.
I have an online question from Andrew Schaefer. Would you argue that the different levels of organization, cells, tissues, organs, organism, can also be recast as subsets of information processing systems that receive and use different cues and signals from the environment? But he goes on. One big question is, how are these information processing systems linked within and across scales of biological organization, and how information processing flows from cell to organs and from organs to cells to reinforce the levels of organization? For instance, cells synthesize ECM, which then reinforces the local environment to perpetuate specific cell types. Did you catch the question at the beginning?
That was a really great comment and great observation.
Yeah.
I think that is one of the questions at the heart of 40CP and definitely something dear to myself. I feel like, Andrew, you probably have watched, maybe have you watched my vision talk for 40CP from before? I think part of what you're also getting at is not just that cells reinforce their own local environment, but in order to coordinate across all these systems, cells need to figure out what information they need to abstract about cells somewhere else, right? If I'm a cell over here, I don't necessarily need to know the exact detailed molecular state and environment of a cell that's way over here. And so one of the questions we need to answer in 40CP is, how do cells across tissues, both within tissues, across a tissue, and across systems, what information do they need to abstract about someone else? Right? If I draw a smiley face, you know that I'm representing a person. I don't need to represent the exact snapshot of the person all the way to the cell over there for them to know, aha, they're trying to send me a signal of a person. And so I think that's another really important question that gets at the heart of what you're thinking about, Andrew.
It's very beautiful and interesting system. So very technical question. So for this, you are using the minimal medium, or the bacteria actually is a factor here? If the bacteria is in the system, how their growth are affected by this crystal?
Yeah, so the way we set this up is that this is in a buffer, and we pre-grow the bacteria. So there's a set amount of bacteria, and the bacteria aren't separately growing. They don't have media for them to feed. So basically you do get the natural food source for the dictya, but the dictya aren't competing with bacteria.
I see.
We haven't set it up such that we put the media that both the bacteria and the dictya are happy in at this point.
Yeah, I think we should probably move on. Thanks, Allyson. And last but not least, we have Shaohe, another fresh group leader at 40CP.
This needs to go to the first slide. All right, so I'm also a new group leader here. So the story I'm going to talk about will be the work I did in my postdoc lab at NIH with Dr. Ken Yamada. So we just mentioned a little bit of extracellular matrix. I'm going to give you one particularly interesting example that why some extracellular matrix might be the key for certain morphogenesis question I'm really interested in. So before I talk more about that, I want to kind of introduce a little bit about understanding different aspects of understanding, because I have been probing different people during my first week here. What does understanding mean to you? And everybody gave me a very different answer. So to me, I think what was particularly interesting for understanding a biological system or process is you want to know the so-called biological implementation of the hardware, what are the components, how they are connected. And that can be done by using the embryonic approaches by perturbations and observations. I think Janelia is particularly suitable for this kind of approaches because we have microscopy, we have FIPSing, we have all kinds of different tools and toys we can play with. And then we can use modeling and reconstitution-based approaches to try to get the operating logic and the insight from it. So by inside, I mean like it's always kind of like a thin line walking between being simple and simplistic, as Ruslan was talking about yesterday. It also relates to the scales, temporal and spatial scales that we care about the system. For me, what's particularly important is the micron to millimeter scales, and from minutes to hours. So I want you to keep those in mind, and I want to like kind of highlight the importance using this story, the importance of using these multiple complementary approaches. So the process I studied is basically, in a nutshell, is how the cells and the actual cellular matrix are helping each other to build organs with certain geometry. A common feature of many mammalian organs is this shared branched structure. So this structure is really important for these organs to function because they allow these organs to package a large amount of epithelial surface area within a compact volume. So the way they generate this beautiful structure is by a conserved development program called branching morphogenesis. And this branching morphogenesis eventually generates numerous buds at the end that's specialized for biological functions like breathing or blood filtration or saliva secretion in these different organs. It's really important to understand the process of branching morphogenesis because first of all, it's beautiful, and really, like we have curiosity about how this like fascinating process is happening. And second, it has implementations for tissue engineering efforts to build the kind of artificial organs, trying to help the patients with defective organs. So the branched organs eventually all have a lumen that will allow the air or the liquid to flow through. But at the very beginning, some organs will have a lumen already, but other organs will have a packaged, densely packed epithelial cell to begin with. So lungs and kidney are the examples of the former, where they have a single-layered epithelial sheet, where it is thought the buckling of this single-layered epithelial sheet is underlying branching morphogenesis. But it's not immediately obvious how a structure with densely packed epithelial cells can undergo this kind of branching morphogenesis. And I'm going to use my favorite model system, which is a mouse embryonic salivary gland, as my toy model to try to answer this question. So I'm going to show you a 4D movie to highlight why this is such an attractive model to study branching morphogenesis and other aspects of organ development. At the core here, there's an epithelium that's a lollipop. And surrounding this epithelium is this mesenchyme. What is invisible here, but very important, is encasing this epithelium that's a basal membrane. When I play this movie, you can see this single epithelial bud is repetitively split into a bunch of grapes. And that process is basically the branching morphogenesis I'm interested in. The mesenchyme surrounding them is providing the necessary growth factors to help the epithelial cells to proliferate and differentiate. So this is a very approachable system because we can do live imaging. We can also analyze at higher resolution at different time points. So my work began by looking at higher resolution of how these cells are moving using 2-photon microscopy. So if you look at the cell movements of the epithelium, it is quite random at first glance. But is that really the case, or is it because we didn't look carefully? I'm going to give you some evidence to say that it's really this surface layer of the cells and the interior cells are two separate populations instead of freely exchanging. And the first key experiment I'm going to show you is this photo highlighting experiment. I'm using this photoconvertible fluorescence protein, KIKGR, which normally is a green fluorescence protein, but you can use blue laser to convert them into a red fluorescence protein. What I did here is I went around a growing epithelial bud and photoconverted patches and then followed their movement. As this movie shows, you can see that most of the cell movements are confined within a very tiny distance from the surface, indicating like these two parts of the organ is really representing two compartments. Another experiment, I used this double transgenic mice expressing a keratin-14 RFP reporter and a histone EGFP reporter that's tracking the nucleus. So by looking at this keratin-14 reporter, I hope you can appreciate on the middle plane, it has this enrichment in the surface layer. When the movie plays, you can see this surface layer that's highlighted by this red reporter is expanding and folding inward, that is splitting one existing bud into more buds. During this observation, one interesting phenomena I found is when the surface cells are dividing, the reason I track this is, of course, I want to know where the new surface materials are coming from. So when they undergo cell division, they will first delaminate from the surface layer, enter this crowded interior, but their daughter cells somehow remember where they come from, and they will always go back. This one movie is such an example where I track one cell back to its surface. By tracking many such occasions, I found all the cells will eventually return, and 80% of them return relatively quickly within about 4 hours. The question comes is, why, what is driving this? And by looking at the E-coherent staining in this tissue, I quickly proposed a very simple idea is it must be driven by differential adhesion-based cell sorting, so that if the cells can remember they are low adherent just by physical cell sorting based on this differential cell-to-cell adhesion, it will be sufficient to push the cells back to the surface. But E-coherent is only one type of adhesion molecule these cells might be expressing. And I'm interested in looking at the global adhesion profiles where I used single-cell RNA sequencing. So by profiling the epithelial cells, we have about 7,000 cells here on this UMAP, clustered into 4 different major clusters corresponding to the surface and interior of the bud and ductal cells. And the major take-home message is really we found this outer or the surface layer of the epithelial cells have a strong cell-to-matrix expression, such as integrin expression, and weak cell-to-cell adhesion strengths, including their E-coherent expression and some CAM molecules. So that gives me a simple model of saying okay, this system seems to be having 2 major cell types and one really important extracellular matrix, which is shown in yellow, that's a basal membrane. And the surface cell type has the combined low cell-to-cell adhesion and high cell-to-matrix adhesion, and that seems to be the key of driving this budding morphogenesis. So the idea here is the proliferation of this surface layer provides the raw material, and this raw material is temporarily stored in the inside, and that is pushed out to the surface by differential cell-to-cell adhesion, and those cells then rely on their strong cell-to-matrix adhesion to become a thinner layer that will fold inward onto the interior layer. And that, of course, raises the question whether these surface cells will proliferate, will grow at a relative advantage than the interior cells to have more cells. To test that, I did mathematical modeling considering a very simplified scenario with a 2 compartmentalized sphere. And then I derived experimentally observable values, and all the data supports faster surface expansion and budding morphogenesis. So that gives us hope that maybe we can then reconstitute this seemingly complex budding morphogenesis by manipulating some simple parameters by engineering the cell adhesion. So before diving into the cell engineering efforts, I want to show you this proof of principle movie, where I isolated the epithelium from the branching salivary gland, dissociate them into single cells, and then we can see these cells are capable to aggregate into a spheroid and then undergo budding and branching morphogenesis. So this is very hopeful, of course, because it establishes the feasibility of from individual cells we can really reconstitute epithelial morphogenesis. I then use this human cancer cell line, DLD1, as the test tube to do some engineering to hopefully reconstitute some part of the morphogenesis. And this is a perfect starting point for us because it's an epithelial cell, and in 3D culture, they do not undergo spontaneous morphogenesis. To manipulate the cell adhesion, I implemented the inducible CRISPR-I system, where a dead Cas9 and the transcriptional repressor domain CRAB are fused to two domains that can be inducible dimerized by the abscisic acid. I then identified guide RNAs that can efficiently reduce the equatorial expression by abscisic acid. So I also marked these two types of cells with two different colors so that I can distinguish them more easily. By mixing culture of these two cell types in 3D cultures, you can see when I add abscisic acid to reduce cell adhesion in this system, the magenta cells will surface. That is based on the differential cell adhesion. But these cells do not spontaneously form a basal membrane, so they need some extra help. After I give them matrigel, plus the induction of equatorial reduction, we can see that robust budding morphogenesis can occur. So that is really exciting for us because by simply manipulating one parameter and giving them some help to form this really important extracellular matrix, we can observe the critical first step of branching morphogenesis, which is the budding morphogenesis part. So this part of the work has been published, but it highlights the importance of using this kind of reconstitution approaches to test the simple rules we might discover from the embryonic system, which is relatively complex, and then try to say, like, this might be the operating principles this system is really using. So here at 4DCP, I'm going to try to use a lot of existing tools and also some of the new tools my colleagues will be developing to further further using this system to ask questions regarding morphogenesis and organ development in general. So the three major directions I'm going is all related by understanding by building. The first thing is how do ducts form and how can we build a complete branched structure, not just a cluster of buds, but with a full branched structure. And the second part would be try to give some function to these bud and ductal cells to make them useful. That can be another testing test tube for achieving the key molecular modules that's underlying the functions of different tissues. And the last one is related to the brain-body people, that is trying to establish an organ-to-body interface. And for that, I want to show you one last movie to demonstrate why this organ explant system can be a really beautiful model to study this. Because we don't know how neurons are recruited by epithelial organs. So we can see from this movie, we can visualize this process. And this is a process of 40 hours total. And we can see this neuronal processes is reaching out and crawl on the buds of the epithelium and eventually seem to stabilize around the ductal region. A lot can be done in this system because we can use CRISPR-based, lentivirus-based CRISPR knockout in both parties of the cells to ask which molecular modules are the most important. And finally, I want to acknowledge my postdoc mentor, Ken Yamada, being a great mentor and very supportive, and one of my lab mates, Kazuma Matsumoto, who helped me with many experiments. The NIH facilitates my collaborators, and my lab website is already up there, so please check it out. We have some opportunities for visiting scientists and undergrad scholars to do some summer program, and you can also follow us on Twitter. And with that, I can take any questions you have.
Yeah, hi, I have like two quick questions. The first one is in the engineered system, you kind of break the symmetry yourself, but I was wondering in the normal environment, do you know what's the cue that causes that symmetry breaking that separates the cells into the outer ones that have lower adhesion and the inner ones that have higher adhesion?
That's a great question. So if I would give it a guess, I would say it's probably the gradient, like they are sensing the growth factors sending from the surrounding mesenchyme, right? So that naturally can give you a gradient that will differentiate that. Another part is the boundary condition, right? So only the surface cells is directly adhering to the basement membrane through the integrin-based signaling. So that could be another factor.
And then do you also expect this to be the case for branching morphogenesis? Because you showed it for budding morphogenesis, but for branching, do you also think that that differential adhesion would be enough to drive branching morphogenesis?
Well, I'd like to clarify. I think by branching here, you mean like really the ductal morphogenesis, right? So I don't think differential adhesion per se would be sufficient. Well, first of all, like we already give them a lot of differential adhesion here, and by observing the system for like much longer time, like they never really spontaneously form any ductal structure. Actually, I think it's a really big open question that is probably bigger than a lot of people, or like more complex than people have thought about, like how, like giving a bunch of epithelial cells, how they form a duct. Right? So we know quite a bit of like what kind of things we can perturb to make them not forming a duct, but we have no idea of like the other way around, how to make something to form a duct.
Beautiful, beautiful work. Do you know if the high integrin, low coherent cells versus low integrin, high coherent cells, whether they differ in laminin expression?
Laminin expression, yes. So I do have some single molecular fish data looking at the expression of ECM proteins. The most striking difference is in collagen IV expression. You kind of like have a necklace of enrichment in the outer cells, and you have relatively little collagen IV expression in the center. Laminin expression have a similar sort of differential expression pattern, but the difference between the two is not as striking as collagen IV.
Thank
you. A question online from Pilwali. Is any parasympathetic innervation of salivary branching morphogenesis and structuring dependent on feeding conditions?
Depending on feeding conditions? I don't know. Yeah, I don't know if people looked at that. I can definitely look at it myself using this explant model.
Okay, let's go with the green.
In the daughter cells that you saw branching off of the outer layer, did you see any kind of difference in classification of like their integrin or E-cadherin? Were they already like predetermined to have high integrin expression, and then that's what drove them towards that outer layer, or was it influenced?
So I think by the time I was analyzing the data, the cells have already kind of like determined, like once they are outer cell, they are like going, their progeny is also going to be that type of high integrin and low E-cadherin. So one of the experiments I did is tracing them in live imaging and then fix them by staining, let's say, E-cadherin. That part is actually in the paper to say like that still expressing very low E-cadherin. Another thing is if you look in the single cell data, I did do some RNA velocity, and the outer cell has this like swirling pattern that's representing their cell cycle phases. And those are not differing very much in terms of their integrin or E-cadherin expression.
I think we should probably cut it off there, but please ask Shaohe during the break. Thank you very much.
So we break until 4:05, so a little over 15 minutes. 15.